This is not working for me. I'm sorry. All right.
We'll just take a moment. Okay. So, hey everyone, thanks so much for coming tonight. Tonight's Popping the Science Bubble Science Lecture Talks. We're so excited. If you're hanging out, feel free to come and listen. We have some really interesting talks lined up tonight. So welcome to Popping the Science Bubble. This is a monthly seminar series that aims to share new research findings from grad students and postdocs at UC Berkeley with the general public and create constructive discussion about a variety of science topics. We have 2 speakers at each seminar who will talk about their current research or a topic that they just find really interesting. The organizers are 4 graduate students at UC Berkeley: Kayla Zong, Daniel Bretthauer, Madison Lemer, and Oksana Andrei. Lectures happen on the 3rd Tuesday of the month at 5:30 p.m. The programs are also archived on YouTube, so you can view them later and share them with others. Popping the Science Bubble also has a website and a listserv that you can join to keep up to date with new Science Bubble lecture topics. So I'm going to step aside and let our team introduce tonight's speakers.
Yeah, so thank you for being here. We're excited for tonight's speakers. As Kelsey said, we host this talk the 3rd Tuesday of every month, and we have researchers from a wide variety of departments to share their interesting research. And so if you're interested in learning more about future seminars, you can join our listserv. We have a QR code, which unfortunately the internet is not letting us download and show, but you can also use bit.ly/PTSB emails. You can also visit our Twitter, Facebook, or find previous presentations on YouTube. And so for tonight, we're excited to have Rumi and Joseph presenting for us. And so this is an open format talk. And so we encourage you to ask questions throughout the presentation. And so you can, for those of you that are on Zoom, you can use the chat function or the Q&A section. We'll ask your questions that you put in the chat there after the talk. And so to start us off, we'll introduce our first speaker.
Yeah. Like most of them just sitting on Zoom. So hi everybody. I'm here to introduce Rumi, our first speaker of the night. Rumi grew up in Fresno, California, out in the Central Valley, but then for college moved out to the big New York City, where he attended Columbia for undergrad. After college, he came back to Berkeley, or California, Berkeley specifically, joined the MCB program here on campus. Outside of lab, he likes playing soccer, cooking Pakistani food, and climbing, as well as just hanging out with his buddies. I'm excited to have him speak with us tonight, so feel free to take it away.
Thank you. Thank you, everybody. So my talk today is to energize or not to energize. And really, we're looking at how our cellular mitochondria can control large global processes of organismal metabolism and also differentiation. Okay, so essentially all multicellular life requires some internal energy production of some sort. When we look at plants, for example, plants can take in energy from light, and then through the process of photosynthesis, they're able to turn light energy into oxygen and sugar or glucose. We unfortunately are not so lucky. We cannot turn light into energy. It would be very nice, but we can't do it. So instead, we have to rely upon a different cellular compartment called the mitochondria, which essentially will take oxygen and materials from the environment, such as sugars, carbohydrates, or fats, break them down, and then form energy in the form of the small molecule ATP. And ATP powers essentially all biosynthetic reactions and is incredibly useful for nearly all action and process that we undergo. So, how do mitochondria generate ATP? Well, like I mentioned, it comes from an influx of our environmental diet. So food that we take in is broken down into its constituent components. So fats, lipids, carbohydrates, and sugars. And then these nutrients are fed into a series of enzymatic reactions here called the TCA cycle, which breaks down the metabolites into a series of, or a few very high energy electrons, which is then fed into a protein chain, ultimately generating ATP that we can use to power any movement or any action. For example, contracting my arm, any thoughts going through my head right now, these all require ATP. So, taking a deep dive into the mechanistic process of how ATP is made. There's a lot going on in this slide, but really the only important thing to focus on is the direction of the electrons. So the electrons are going from left to right across different protein complexes. And ultimately, these electrons are depositing energy with each step they go to the point, or the terminal point, where the potential energy from the electrons is turned into ATP. So essentially, it takes energy to make energy. The energy contained within these electrons, which comes from our diet, is then used in combination with hydrogen and oxygen to make both ATP and water. The main takeaway is that electrons from metabolic breakdown power the synthesis of ATP. Now, there are certain cells that are very dependent on mitochondrial ATP production and thus energy.
Yeah, can I ask you a question? I'm just curious, where are the hydrogen atoms actually coming from to make the gradient? Like, so you're passing these electrons, but it's not like the molecules from the electrons. Where's the hydrogens coming from?
Right.
Yeah.
So where are the hydrogens coming from?
Yeah, where are the hydrogens?
Yeah, so essentially mitochondria have pores within them that allow molecular oxygen and hydrogen to flow into the, both into the matrix, but also into the intermembrane space. The matrix is below, the intermembrane space is above here. And the hydrogen ions are then pumped out as a result of the electron transport chain and then brought back in during the final step of ATP making. Molecular oxygen is brought back in as well, but it's also all around this space, just not shown. And actually, it's a great question, something we'll get to later. What happens with oxygen at various other steps during the electron transport chain? Any other questions? So, can anybody suggest or know of a cell type or tissue that's very dependent on energy or requires a lot of mitochondria? Yeah.
Neurons?
Neurons, exactly. So I have a few examples here. For example, muscle tissues. The act of contraction requires ATP. Neuronal tissues, for neurotransmission or to send a signal, this is an ATP-driven process. And heart tissues are some of the most intensive tissues when it comes to mitochondrial activity in order to keep beating and maintaining blood circulation. Now, given that mitochondria play such an important role in three major cell types, including, but essentially all cells, mitochondrial dysfunction is a real driver, a really potent driver of disease. And in particular, it can affect muscle cells, cardiac tissues, and neuronal systems, in which for cardiac tissues, heart failure can be driven by mitochondrial dysfunction, neurodegeneration, such as Alzheimer's, Parkinson's, and dementia is rooted, or at least partly rooted, in mitochondrial impairment, and muscle diseases such as, or muscular diseases, excuse me, such as muscular dystrophy are linked to aberrant mitochondrial activity. So it is really crucial to understand when mitochondrial activity is important and are there steps we could take to ensure that mitochondria are functioning at a healthy level. So to summarize so far, our body is filled with cells and each cell has several mitochondria. In healthy cells, cellular fuel in the form of fats, sugars, and carbohydrates enters and it's created or turned into energy in the form of ATP within mitochondria. However, damaged mitochondria are unable to import as much fuel or unable to make as much ATP, and they're not able to meet the energy demands of the cells. Therefore, diseases that people already have linked to mitochondrial dysfunction will get worse.
muscle?
No, so it really requires a differentiation cue or a switch. So in our case, what we do is we switch to a low serum or a low nutrient condition, essentially, and this gives them a cue to differentiate, essentially. Yeah, so we switch to like a low serum. Essentially, we reduce it by about 10-fold.
That's interesting because I thought it would take more energy to make the muscle so light.
Yeah. So mitochondria, it's somewhat paradoxical in that like mitochondrial output is increasing during this process, but essentially for this cell line in particular, I don't know all the fine details, but the initial cue is to starve them for a bit and then they differentiate. But mitochondrial output does ratchet up during this differentiation process. You're essentially burning through whatever reserves you have in order to make these differentiated cells. And, what we're able to observe pretty, strikingly is that when you when you have our control cells, you're able to form muscle myotubes in green. But when you have a condition of perpetual reductive stress, you're no longer able to form any larger muscular structure. And when you have ROS scavenging, essentially removing all of the ROS from a cell, this keeps the cell in a state of reductive stress as well and results in no muscle myotube formation. And this had us wonder if reductive stress is so deleterious for such an important process such as muscle cell differentiation. Cells must have evolved a way in order to counteract this reductive stress. How do you go from a period of reductive stress to then potentially reactivating mitochondria in order to deal with reductive stress? Is there a cellular response in cardiac tissues or in muscle tissues, in this case, for cells to overcome a period of reductive stress? And this is a bit of a broad overview, but our lab was able to find out that there is, under sufficient mitochondrial ATP production and electron transport chain activity, ROS levels are at either steady state levels or upregulated. And this leads to stabilization of a mitochondrial gatekeeper protein called NIP1. NIP1 is at the outer mitochondrial membrane, and it controls ETC activity. It essentially regulates the influx of metabolites into the TCA cycle, which then feeds into the electron transport chain and then leads to ATP activity. However, under conditions of low or reactive oxygen species, by proxy, cells or mitochondria are not activating the electron transport chain, and you're undergoing reductive stress. Under reductive stress, NIP1 is degraded by the cell, which results in mitochondria being turned back on and reactive oxygen species being upregulated, and consequently, in parallel, ATP production being resumed and upregulated. Using a mitochondrial reactive oxygen species marker, we're able to see that in control cells, in muscle cells, mitochondria are producing decent or physiological levels of reactive oxygen species seen in white. However, when you lose the cellular factor responsible for degrading NIP1, you get very little to no mitochondrial reactive oxygen species because NIP1 is stabilized at this point. It can't be turned over or degraded, even if you're undergoing reductive stress. Loss of NIP1 leads to production of mitochondrial reactive oxygen species, but and what was really nice is that loss of NIP1 and loss of the NIP1 degrading factor restores or rescues the condition back to physiological levels of reactive oxygen species. So even though you can't degrade or remove NIP1, you don't have any NIP1, so cells essentially restore mitochondrial activity. So to conclude, cells have a mechanism in order to deal with reductive stress. known as the reductive stress response. Through the reductive stress response, cells are able to turn back on their mitochondria. They're able to boost levels of ATP. I'm not showing all the data here, but they're able to restore ATP production, boost the oxygen consumption rate back to physiological levels, and essentially rescue or partially rescue deleterious effects of prolonged mitochondrial inactivity. Again, prolonged mitochondrial inactivity can lead to conditions such as cardiomyopathies, diabetes, tumorigenesis, and is needed to be restored back to physiological levels in order to proceed with tissue differentiation, such as in myogenesis or the formation of muscle myotubes, cellular growth and proliferation, and metabolic biosynthesis. With that, I'd like to thank my PI, Professor Michael Rocca. I'd like to thank all of the Rocca Lab members for being such incredible supportive colleagues. I'd like to thank the Berkeley MCB graduate program and the Howard Hughes Medical Institute. On one last note, for those who are interested, I'd like to recommend two scientists whose work I find particularly exciting and informative. Professor Marcia Haggis at Harvard Medical School, whose studies focus on mitochondrial function in aging and age-related diseases, and Professor Navdeep Chandel at Northwestern University, who has really characterized reactive oxygen species not just as byproducts of the electron transport chain, but as second messengers, and has really fleshed out their important signaling functions, which is something we try to do as well. And with that, thank you for listening, and I will take any questions. Any questions in the chat?
I'll give them a minute to plan.
I have what's the long-term effect? And the long-term effect being studied for the loss of both the NIP1 and NIP1.
NIP1 and yeah. So basically, it's the NIP1 degrading factor and NIP1. So NIP1, if you lose it, actually, it has been shown that at least in mice that they develop abnormalities later on in life, and that their muscle tissues are just essentially don't function as well. The degrader of NIP1 is called FEMLIN-B, and it's there's actually a developmental syndrome linked to hyperactivation of this degradation process. So if it actually can remove NIP1 too quickly, that's bad for the cells as well. So you really need to be able to time the reductive stress response and also know when to shut it off. If you let the reductive stress response go on for too long, that can cause problems of its own and actually cause oxidative stress potentially.
And what's the long-term effect for losing both of them? Like,
yeah,
propose as a treatment or some kind of restored function, but what's the long-term effect? Because actually, it's not a natural state of a cell, is it?
Yeah, so like I'd say the long-term effects we really don't know. It hasn't been well characterized. I think it's something our lab's going to try to do in the future, hopefully. But again, you need to do beyond mouse work. You need to do larger patient studies, and I don't think a lot of people have done this yet. But it's certainly something we're looking at, as well as seeing, going through patient and genetic data to see how these things track across diseases. Yeah.
And are these effects very local, or can they spread?
I wouldn't say they can spread, but I would say like tissues depend on each other in order to function. And in particular, some of the problems with, or some of the mutations in NIP1, for example, have been associated with diseases of the heart or nervous system issues. So certain tissue types in particular are probably more susceptible. Yeah, thank you.
Yeah, maybe on the other side of things, so you mentioned, when this thing, the, stress response failed or whatever, like the degradation products and stuff, have those been well characterized about what they're changing about the actual signaling proteins? That's like making it so they can't like some sort of more mechanistic information about what's actually happening.
Yeah. We've published on that recently. It's a pretty interesting mechanism that this occurs at mitochondria, and really it's the NIP1, for example, has cysteine amino acids that are kept in an inactive conformation, so it can't be recognized under oxidizing conditions. But when you reduce the protein or you're in reductive stress, these cysteines form a bridge with the degrader protein. So only under reductive stress can this protein be degraded. And it occurs through a zinc interface. So both the degrader and NIP1 bind through a metal, and this can only happen under reducing conditions.
And so what's when this overactive degradation protein, like how do they overcome that? 'Cause I'm assuming that's not just like a super bad environment all the time.
No, it's a great question and something we're actively looking at right now. So it's a graduate student project in the lab.
Maybe one more follow-up. Where does it actually interact? Maybe I missed this, but where does it actually, like, where's FINF1? Does it interact actually with the mitochondria itself, or does it interact?
Yeah, it actually is. It's held by an outer membrane complex. Okay. Yeah, and so the degrader is held there also. NIP1 though can move. It actually can move between the lysosome and the mitochondria. The degrader protein is always kept at the mitochondria.
So you I didn't realize actually the cell can have several mitochondria. Does it pretty fixed? Like what type of cell has how many mitochondria?
So it's a big scraggly network essentially. It almost looks like I'd say it looks like a woven fishing net in a way. It's really spread out across the cell, and certain cell types that require a lot more mitochondrial activity will have many more. There's no I don't exactly know a number off the top of my head, but for example, as I mentioned in the talk, like cardiac muscle, up to a third, if not 40% of the cell can sometimes just be occupied by the mitochondrial network. But other cell types that aren't requiring mitochondria as much might have much lower levels or a smaller network, but it's this tubular network of sorts that is interconnected throughout the cell. This is so every single part of the cell can get quick and easy access to ATP.
And they form kind of fixed number when the cell first split, or they can grow or copy themselves, or
they can't. So mitochondria can split off and fuse, or it's a really good question, and actually like a study that came out fairly recently showing this. They can fuse by themselves, and they can divide by themselves, and this isn't tied to the cell dividing. It happens independently of the cell dividing. Yeah. Of course, thank you.
All right. Well, thank you so much, Rumi.
Thank you, guys.
That was a great talk. Oksana will introduce our second speaker of the night.
Can I have one?
Yeah.
Just because I had my QR still screen sharing.
Nope, please go ahead.
I'm going to go ahead and fund it.
I'm in that. Yeah.
Our second speaker tonight is Joseph, who grew up in North Michigan. He went to Michigan State University, where he studied physics and mathematics. After graduating in 2020, he started graduate school as a physics PhD student at UC Berkeley. He has found single molecule biophysics and its implications for applied physics particularly intriguing. This has led him to his current research on observing and modeling molecular motors. Outside of research, Joseph enjoys pickup sports, does sound mixing for live concerts of Bay Area music acts, and tries to write in his spare time. We are very excited for your talk. Take it away.
When I can do the presentation.
Never that.
Yes, exactly. I think right? Yeah. Okay, I'll share it and then
Yeah, that one from you. Yeah. Cool. Perfect. Thank you so much, of course. Yeah. Thank you for the introduction. Thank you, Rumi, for that. That was an excellent talk and surprisingly an amazing segue for my research. And also your question also segued into my research. Is this going to be okay right there? Yeah. Oh, it's probably not good.
Get rid of that. If you just go to more word.
Oh, more. Oh,
Wait, move it.
right.
Yeah. Or then hide floating mean.
There we go. Maybe that's fine. Cool. Okay. So yeah, so I'm Joseph. So I'm a actually physics student, but I study biological problems. And specifically, I'm interested in how our cells deliver microscopic cargoes, which very fascinates me. So in the top, hopefully you'll understand by the end of this presentation what this movie is showing. But small spoiler, it's an experiment that I've done. And then on the bottom is not an experiment I've done, but rather an animation of kind of, you know, a trajectory of how maybe I sometimes think about these things as just little cars driving on their little microscopic highway network. So, so some takeaways I kind of want to outline for today. So one is biomolecular motors, which are these things that actually produce energy, or don't produce energy, they take energy that was produced by ATP, and they use it to actually make directional transport, are important to maintain cellular life. We also, you know, second lesson, we can actually study what affects these motors, and by reconstituting the system, not in the cell, but actually outside of the cell. And then this gives us like optimal control on testing different hypotheses. And three, there are many different ways in which motor transport can actually be regulated to complete a delivery of cellular cargoes. So without further ado, I want to introduce our main movers, our motors. So these are cars or vehicles that move on microtubules. So many of you might be aware that the cellular life is not a very equilibrium process, meaning that we don't just like kind of sit around and just let chemical processes take place. We usually have to drive them in some way. And this is one of the most direct observations of that driving, is that you can actually see motor proteins such as this kinesin, which was discovered about 40 years ago now and has famously been named to this great gif of this little kinesin walking hand over hand like a person on a microtubule lattice. And these kinesins actually walk in one direction by consuming one ATP, so the ATP that mitochondria produce per step. And so they just walk along this microtubule lattice in one way. And then there's also other cargoes that move the opposite way called dyneins. This is a little more complex. I don't have a movie of it, but basically the dynein, if kinesins move it this way, dynein's going to move the other way, kind of like a train. And they basically move both by hydrolyzing ATP and moving cargoes in the cell. But of course, this doesn't seem very productive because, well, why couldn't I just let my cargo kind of just sit around and do whatever? Why do I need to consume energy to move things? Well, it turns out that a neuron, where this is a very, you know, cartoonish picture of a neuron, is actually very long. So a millimeter can be a neuron. So, but, you know, and as we talked before, is a high consumer of ATP. And there are even neurons that are actually a meter long, a single cell, that are very long axonally, that stretch very long distances. And if you were to say split, let's say you want to produce ATP, you want to be able to give cells access to a charger, a mitochondria, so per se, which Rumi kindly introduced for me. Well, if you wanted to have that mitochondria just split from one of them and then just move over to the other end of the cell, it would take on the order of a month to just do that a millimeter. That is how just randomly like bouncing around would work. And as you can imagine, this would be the same thing as your Amazon package coming to your door. If they just had an Amazon package and someone just walked around randomly, there'd be no way that it would actually end up to you in your lifetime. So what we need is we need a train system. We need these motors to be able to take these cargoes and actually bring them to the places they need to go. And so when this transport network fails, there's actually a lot of diseases that come about, especially ones related to the brain, such as Alzheimer's, Huntington's. There's also, I didn't put on it, but like smooth brain disease. All these things come from different parts of the intracellular transport network failing. And so we really do need to kind of study this to try and understand how, because it is actually a very open question in biology, as how I can take a single protein and actually be able to deliver an actual cargo. So how does the motor know where to go? Because as far as I'm concerned, most chemical reactions are local and they don't usually see something super duper far away from them. And super duper far away in this case is, something on the order of 8 micron. So in order to do this, there's actually a couple major theories for how intracellular transport. So I should make sure intracellular means within the cell. We're not going between cells in this case. How is intracellular transport actually regulated? So it all has to deal with, if this is my motor, if this is my thing that's producing ATP, or consuming ATP to be able to produce work, then there are things that it interacts with at any given time that could possibly regulate its function. For one, the microtubule, the thing that actually walks on. So the road could change, right? If I have a different road, maybe then, if I had a train track rather than a, highway, I'd have different things that would be able to walk on that certain pathway. There are also microtubule associated proteins, so just other proteins that exist on the microtubule, but represented by this little cartoon over here in that little green spot. And these, are actually they're helping to stabilize the microtubule. It's actually not a fixed road. You need like regular updates, and you need regular repairs to it. So these maps actually, which are called maps for short, are actually kind of like road architects, and they exist all the time. So motors constantly have to interact with them. And then finally, there's also motor cargo adapters. So like the cargo, which happens to be a cartoon of a mitochondria in this case, could somehow has to interact not to the motor itself, but through a cargo adapter, something like a hitch. And so this cargo hook could change to only grab onto the packaging you need. If you only need to transport a small box, you don't need to host a big truck, that sort of thing. And that would be, the cell's way of doing that. So, and there's actually a lot of different theories, and there's a lot of different ways we can test these, because this is a lot of moving components. So this is something very hard to do in a cell, right? In a cell, there's so many of these things just existing around. It's very hard to differentiate what's important, what are specific things that could be changing. So in order to kind of hone in on this sort of large question open in biology, we try and specifically look at just specific interactions. So just look at the motor with the microtubule, or the motor with the map, or the motor with the cargo adapter that we're interested in, and see how that could modulate our behavior. So we will produce a bunch of proteins that, are our motor, or we can do that outside of cells, which is a lot of fun. And then we purify those proteins, and we can actually we can label them fluorescently, kind of as Rumi showed. There's cool movies of labeled proteins. And then we can hit them with a laser. And when we hit them with a laser, we actually take advantage of the idea that we can excite these particles. And not only can we excite, we can not only just excite, a bunch of particles and a lot of ones that are floating around, we can actually only excite particles very close to the surface. And we do this by a concept that maybe you all might think about from if you've ever kind of if you ever like looked up from being under a pool. If you look straight up, you can usually see outside of the pool. But if you start looking to the side, you suddenly cannot see the exterior of the pool. And this is because the light that you are trying to see, the optical path, cannot go through from the air you cannot go through from your water to your air. Sorry, it's the opposite way. But anyways, so the idea is that if I were to look at a piece of glass, I sometimes get a reflection as well. And we can actually take advantage of this reflection to totally reflect our laser just only at the surface we see stuff. And so then we can get you know, here's our cool little laser setup with many different cool lasers. And we can get cool movies where we now take our microtubule and our proteins that are labeled, we put them on a glass slide. And then when we put them on the glass slide, we get to see a cool movie, which would look like the one before, but for some reason it's loading. So let me, whoop, maybe if I escape. I broke it. But so the idea would be is that what you would see is then every one of these little motors that land on a microtubule, you would watch them walk. You would be able to see their little excited little laser lights. They're just little dots moving around. And you can watch them walk on a microtubule. And then from here, if it will go on, you have any tricks for me about how to end? Okay, maybe let me just end it. No, that's end meeting. I don't want to do that. Cancel. Stick it up. A way to just stop the presentation.
I'll share.
Yeah. Okay.
Give it 11.
All good. I realize I have a couple movies in here, so it's probably not the easiest thing. Hey, there we go. Do that. Oh, that. Yeah, that's fine though. We can, as long as it can move. That's the only thing that matters.
Yeah.
Can we just exit out of it and get ahead to the next slide?
Yeah.
And there are also technical difficulties.
While this is going on, so how many motors are you seeing at once, like when you take these videos?
A lot of motors. Yeah, so that's a good question. So each of these motors is like about maybe 10 nanometers, give or take. And then we can image a whole field of, so that movie that I was showing is, you know, a microtubule that's about like 10 microns. So basically about 1,000 motors in that one spot. Of course, we're not seeing 1,000, we're seeing about the order of 20 to 30, because if we had 1,000 motors on the microtubule, it just look like a line, and you wouldn't see anything moving. You just see like flowing currents. But yeah, so actually we're seeing a lot of motors at once. And so yeah, it's really cool to think about those, that little picture I showed you, right? It does look like kind of like a car, but a car is on the order of magnitude of a meter, and these things are on the order of magnitude of 10 nanometers, so like 8 orders of magnitude lower than than your actual car, but it still looks like the same thing. Yeah, go for it.
I'm also kind of curious. You mentioned that you have this kinesin, I think it was called, that goes one way, and you have a dynein.
Yep, you got it.
That was going the other way. I not just, pick up a kinesin and turn it around and then.
That's a good question, because you actually want to have, so like evolutionarily, it would, so it's, I guess, let's see if I can tell this right. So basically, the motor itself is hardwired to have a specific way of going. Because if you wanted a motor that could do both, then you'd have to have some sort of switch that would turn it like on or off. So instead, to make these things super efficient in one direction, you kind of have to like put all the gears in one way. And so that in the way that the protein does that is it has to have a certain conformational change that only pushes it forward. Because if you could have it go either way, then you'd probably just have it would just go back and forth all the time, and it wouldn't actually direct something. So you need something that can like put this thing into a gear. And unfortunately, I don't know of a motor that actually switches, whether it goes back and forth like efficiently both ways. And you can maybe like an analogy would be like a car, right? There's only one reverse gear, and there's like 5 different forward gears, right, to make you go faster and faster. So, but it is kind of weird, interesting. Evolution worked out that way, where it wanted 2 different motors to go in each way, and it didn't want it didn't want one motor that could somehow do both.
Yeah, and it also looked like the dynein was like a lot more complex.
Is
there a reason for that?
It's actually, so surprisingly, dynein has a lot more roles than kinesin. So kinesin is highly special. Like kinesin is kind of specialized. There's a lot of different subfamilies of it. Dynein, there's only one major cytoplasmic dynein. So it kind of has a lot more, but it has a lot more function. It has a lot more things that it does, even in like the cell life. But in reality, dynein actually existed before kinesin. So dynein's actually a more ancient protein used, I think, in yeast, but kinesin does not exist in that. So it's very, because it was made to basically, if we think about mitosis, when cells split, dynein actually is part of the thing that pulls those cells apart. So yeah, surprisingly, nature does not do the simplest thing first every once in a while. It finds something that works and then it tries to keep working on that. So yeah, those are good questions though.
We are still having some technical difficulties.
All good. Bear with me. All right. Hey, no one sends back basically. Maybe.
Laptop, if that would be helpful.
No, I think it should be okay.
Okay.
Do you think is this on my drive that's giving you problems?
Maybe, I don't know. You got it back. I think I should be able to get it. 2 minutes, guys, and then we'll have it.
Yeah, no problem. Any other questions? Discussion's great though.
How long does it take for it to from protein from one end of neuron to the other end? If it's 1 meter.
Perfect. That's a good question. So if a motor protein can move at about a micron per second. So basically it would take about 1,000 seconds to move a millimeter. 1,000 seconds is about half an hour. So it still takes a while, but it's much better than a month. And then if you wanted to do a meter, you have to multiply that by another 1,000. So it would be half. So I'm trying to do mental math. So I guess take half an hour, multiply it by 1,000. It would be on the order of like a day. No. It would take, sorry, my order of magnitude math. It would be a month. Yep, you're right. So it would take a month if you wanted to go from one way to the other.
But you have many of them. How many of them are there?
How many of what are there?
The movers.
The proteins? There's hundreds of them. It's not thousands or tens of thousands in a single neuron. But these things, it's not like more motor. Yeah, so it's not like one motor has to bring it all the way there. Just like you have kind of shipment hubs. You don't have one truck bring all your stuff from the one side of the country to the other. So.
And once they reach the other side, what happens to the motor?
That's a good question. So motors will often reach the other side, and then so if they reach the other end, I'll talk about it maybe if we get the presentation back up, but they actually can be transported back. So they might move into an inactive conformation and then get transported back by the other motor. So they can actually tag on as cargoes themselves, and then they get put the other way. So. Yeah, so that might be another reason for why you need two different types of motors, right? More easy to regulate which one's on or off via cargo adapters rather than some cell signaling. Let's see if this breaks it, because there's more movies. I should have included so many movies.
I think it should be okay.
Yeah, I think we're good.
Share it again.
Yeah, let's do it.
All right, and then before we restart the presentation, there's also a question on the Zoom Q&A. Joseph is asking, does laying down memories in the brain involve similar transport motors? And I'll preface that with, I think laying down memories means kind of creating memory circuits, which are usually neuron signaling. So does neuronal signaling involve these transport mechanisms?
Yes, that's a good question. And the answer is there's been no known connection for that. Usually signaling is done by electric signals. These are very molecular motor proteins. They're more important though for sustaining the life and the structure of the neuron. So in ways, these components do contribute to the neuronal structure, which could contribute to that, but they're not a direct link. You are really relying on electric pathways for sort of neuron signaling. But there are a lot of people who are going into, how the structure of a neuron actually changes, how your neuron structure grows, which would then in turn probably contribute to memory, but memory is a very abstract thing still in the science. We're really working on it. I actually saw a really cool talk about it yesterday, but anyways, we digress, but good question. So yeah, a good preface. I'm sorry if there were any of these questions, I was supposed to repeat them for the audience. Hopefully you guys could hear them though, but hopefully they made some sense. So basically for some examples then, so here's some movies. So we were asking about to see movies and here they are. And then to make it easier to kind of visualize the movie, I also spread it out as like a time projection. So basically every one of these dots corresponds to one mover. And if it's moving really like vertically, if it's moving really horizontally, that means it's moving really fast. If it's moving like, if it's not moving at all, it's just stationary, it's just stuck there. So in these cases, what we're doing is we're actually reconstituting all of our system and we're putting in only the components we want to study. So in this case, it's a microtubule, and then we put in our kinesin motor in this case, and then we put in a single map. So, and we put it in at different concentrations. So basically we're just increasing the number of maps on microtubule. And so in a way you can think I'm walking, you know, without anything in my way. And then all of a sudden I'm walking with more things in my way, like in a crowd or, you know, with a bunch of obstacles. And then I'm walking with a ton of things in my way. But what we find interesting is that, you know, usually if there's more things in your way, you would just expect to slow down, right? You have to get around those obstacles, you have to do something else. But there's actually a known microtubule associated protein that actually enhances the kinesin processivity. So in this case, we see that there's actually an increase in the number of motors in moving. So this is like the same number of kinesin motors all around, but then more of them get recruited and start activating on microtubule and get walking. So this is one of those evidences that we could possibly control where motors go by controlling what microtubule associated proteins are on the microtubule itself. Another option is, and then this is that data kind of taken up where it says, as we increase our microtubule associated proteins, which are just these like long strangly guys, that we actually see that the kinesin, you know, number, their run length, so how far they go, and they actually increase, but then at some point they actually go down because there's too many of them, as well as their velocity decreases, which leads to some sort of idea that, you know, as this kinesin motor moves, it's interacting somehow with this microtubule associated protein, which is actually helping it until there's too many of them and then it can't walk. This is still an active area of study, but, you know, there's a paper that I've cited that you could read if you wanted to. There's also cargo adapters. So I talked about cargo adapters possibly being a way to regulate these things. So in this also recently published paper, our group basically took kinesin and dynein. So we took both of the motors. One of them moves positive way, so the positives, you know, to your guys' right, and then minus way, where they say, yeah, kinesin moves to the right, dynein moves to the left, my bad. And what we could see is that when we put in our kinesin, that's this random thing, dynein, with a random cargo adapter, track in this case, which is just one of the cargo adapters, one of the many cargo adapters, what you find is that when there's no MAP7, so that same microtubule associated protein, you actually see that dynein is always winning. So dynein is just there, and it is going to just transport everything. And it actually doesn't even let kinesin bind to it. You can see that there's no kinesin in our track, kinesin in our chondrograph. However, if you increase the MAP7 concentration, you see that now kinesin gets activated. It can move towards one direction and it can bring dynein along with it. So dynein is just chilling there, and dynein just gets inactivated and it gets pulled in the positive end direction, which of course we would assume that dynein doesn't move that direction, it moves the other one. So it's actually a cargo, it's a cargo in itself, and kinesin's carrying it around. And so this kind of leads to this. This is just once again summarized figure result, which aren't nearly as nice as the pictures. But basically it says that if I have no MAP7, then only I see dynein. And if I don't see dynein, then it's kinesin that's winning. But as I increase MAP7, I actually see dynein exist as a cargo itself. And yeah, so hopefully I kind of convinced you of some of those takeaways I told you before, which was, you know, motors are important to maintaining the cellular processes that we were talking about. The diseases come without them. We can also study what affects motor behavior by reconstituting this outside the cell. We're just basically specifically adding only these things, and we see how only those components affect the system, which is very important if you want to do like a ground up model where you want to introduce these things slowly rather than trying to study it from the top down. And then finally, there's many ways, and I just showed you two of them, in which motors can be regulated and to complete a delivery. So basically, these are potential ways that we could start considering how motors can be regulated. Of course, you might say, well, you didn't tell me how I got from point A to point B. And I recommend in that case to be able to explain that. And yep, so acknowledgements, the Yildiz Lab, great. Also, I mentioned some work done by John in the group, who is this guy, and Merva, who did the cargo adapter stuff. Both great other grad students in the group. And if you had any more, resources you wanted to see, I encourage you to check out the Yildiz Lab website, where we have a bunch of these sorts of papers related to intracellular transport. But also, I think the best motor protein explanation is done by Ron Bale. It's a YouTube video from his stuff with HHMI. He's an HHMI guy. He's actually the guy who discovered kinesin back in 1985, I believe. So he has a great talk about how motors work, kinesins, dyneins, all that sort of stuff. So I'd encourage you to start there. There's a lot of cool stuff in these motors. So cool. And with that, I'll take any questions you all have. Questions? I know we answered a lot of them mid-presentation. Hopefully some of them made sense by the end.
No, I'll start us off. Are there, do these cargo or motors transport the same cargo in each direction independently, or are either of them known to only specifically carry unique cargo?
No, that's a good question. So there are some cargos that don't usually need to go in a certain direction. So for example, there's forgetting the name. I think it's okay. So basically, yeah, so when lysosome, am I gonna mess this up? I don't want to mess this up in front of the audience, but basically, if you all know more than me, but there's when a protein needs to be degraded, basically, they're enveloped by there's some sort of phagosome that encompasses that protein, or all those bad organelles or bad proteins, and those usually seem to move very specifically in the -n direction, because the cell body. So the different directions of the cell, there's one that moves to the plus end is usually associated with the exterior of the cell, and the minus end is usually associated with the interior of the cell. I didn't explain that, and I apologize for leaving out that information. But so the dynein seems to be the one that interacts most with those sorts of proteins. Of course, this is through some sort of adapter, which then has to, only take dynein. But in those cases, you have a little bit of a bias towards only moving in one direction or not. But like I said, the cool thing about this is that the molecular system kind of works a lot like how we developed our transportation system. We only really have one type of road, right? But then we have a bunch of different types of cars. And then if we want to transport different things, we have a bunch of different types of things like hitches that we can put into that. So it's very interesting that, it starts from a basic infrastructure and builds up from there to try and get super diverse in terms of what it's trying to do.
So using that analogy, when you have a large cargo, we use a larger truck. But in cells, do you use either a larger adapter for larger cargo, or can you use multiple adapters at the same time to achieve more of a multivariate approach?
Yeah, so in reality, the cargo adapter, so there are cargo adapters that actually take multiple motors. So cargo adapters can take one motor, two motors, some of them can take more. But in addition, the truth is that actually the cargo itself kind of has multiple vehicles usually attached to it. So oftentimes, say a mitochondria is not just actually carried by one motor. It often has multiple motors attached to its exterior, just kind of like how you're saying with your proteins. They just kind of attach to the exterior, and then when one comes off, another one can come on in its place and keep it moving, right? So that's that's a process that we tried to explain actually in this cargo adapter paper, which was that they tried to explain how the motors don't just work as a team. They don't just work as a team, but how they actually know to let go. Because eventually you want that mitochondria to stop where it'll produce energy. And there's actually in cellular evidence that it stops near calcium channels. So near where it would be able to get the most ions to be able to help it with its production of ATP. And so that's a big question is trying to stop this thing as well, rather than just keep it going. That's also kind of a point A to point B question. I don't know if that quite answers your question. Basically, there's a lot of usually more than one vehicle per cargo, which is kind of counterintuitive to how we do it, where it's usually multiple cargoes per vehicle. Yeah, go for it.
So I'm also kind of curious, is there an equal amount of kinesin and dynein in each cell? Does it vary by each cell? Is there more than one than the other?
I actually do not know that off the top of my head. That's a good question for me to know. But I cannot say the stoichiometry exactly. I do know that there's a lot of dynein in the cell. But dynein has a lot of different functions. And so does kinesin. So yeah, I'm gonna say I don't actually know the stoichiometry of those. Like good question I should answer. I should try and answer. But stoichiometry is always an interesting thing to ask about in cells. People often ask me when they look at this figure and they say, "Oh, well, it looks like you have a maximum kinesin. Like, is that what it is in the cell?" And I say, I don't know, because it's like very locally. It's a very spatially incoherent answer, right? You have to look at a bunch of different parts of the cell. It might be different concentrations in different parts of the cell. Our concentration also is different than the microtubule concentration. So it's all these sorts of questions that, I don't feel prepared to answer. That's part of the abstraction we get from in vitro that we can't always bring back to the work. Yeah. So, cool. Other questions?
So this only happens only exists in neurons?
No, actually, there are kinesins and dyneins that exist in almost all cells. So neurons are a good example though, because they're full of that. They require this sort of transport. But there are actually, like I said, dynein exists in most cells because most cells need to be able to reproduce, and they are in charge of dividing the nucleus. Actually, that's one of the things they do. They help with spindle hole formation. Also, kinesin 1 is very common in eukaryotes, but I should segment this this is common for eukaryotic organisms like us. Things like E. coli or like bacteria don't have this sort of cellular structure. They don't actually require transport because they are small enough. They don't have any of these sorts of. They don't require this transportation network to be able to sustain life. So yeah, but good question. Cool. We are over time, so.
I think we're okay.
Do we have any more questions from you?
I have one. It might be somewhat naive.
Do it.
But
I love naive.
Considering you don't want to just like clutter up this road with a bunch of motors, right? But it probably is helpful for them to be somewhat near so they can attach if they need to. Is there, keep up the analogy, some kind of like kinesin garage? Or like known location where they kind of just wait to start transporting, or are they just generally puttering around waiting to join the transport?
That's a good question. And the answer is I'm not gonna say for certain, what's happening in the cellular environment. There's like, actual kinesin garage. That would be cool. I would love to see a kinesin garage, that would kind of say. But in reality, the fact of these motors is that a lot of them are actually auto-inhibited. They don't actually want to walk until they encounter something that requires them to walk, which makes sense, right? If I was a cell and I'm just consuming ATP all the time with dead motors, that is kind of inefficient. So what actually happens is that I think that your garage is actually just solution. Your garage is just motors are floating around, and then they'll eventually attach to a cargo, and they'll either attach to a cargo or they'll get recruited by these sorts of microtubule proteins. And then in that case, they are then able to actually latch on and start moving. Like you kind of have to, the key is more like the cargo rather than the actual motor itself, which I guess makes sense. We're cargos and we have to turn the keys to turn on our cars. So, I would argue that also makes sense in my analogy. But we do keep our cars in the garages, but that's not the cell solution. Cool. That's awesome. Good questions. Thank you very much.
So much.
No, thank you all. That was great.
One more round of applause for both speakers. All right. So thank you very much, both of you, for these fantastic talks. And so if you enjoyed hearing these talks and are not currently set up with our listserv, and you can get notifications about our future talks, you can join our listserv using a QR code that I think will be popping up momentarily. Or you can follow any of our social media accounts, Twitter, Facebook, that we mentioned previously. Here is our QR code that you can scan. We'll also be uploading this seminar to our YouTube page where you can find this as well as previous videos, and we'll send out resources in a post-seminar email from that has been given to us by our speakers. And so we'll be back next month on, I believe it is October
17th.
17th. 17th. And until then, let's thank our speakers one more time for a wonderful talk, and we hope to see you on the 17th.
Yeah. Oh, it worked this time. Yeah. Got the introduction slide.
Thank you so much.
Thank you.
The audience too.
So one, I actually had to like what? So when you're growing these things, you just like put all these, what's it? Myoblasts? No, myoblasts. Myoblasts. It's just like
I think
you did?
you-
Yes.
the web-
Uh-huh.